Good afternoon, everyone. Thank you so much for taking time out to join us for what promises to be a very engaging, interesting, and informative talk. So many thanks again for joining us for our monthly webinar series with the critically acclaimed Professor Kissinger. The talk will be titled Challenges and Opportunities for Omics of Infectious Diseases. Just a few lines on her biography. Dr. Jessica, or rather Jessie Kissinger, is a distinguished professor of genetics and director of the Institute of Bioinformatics at the University of Georgia. Her research focuses on the evolution of parasite genomes, in particular, the genomes of organisms that cause malaria, toxoplasmosis, and cryptosporidiosis. I hope I said that right. Her research area has necessitated the development of new computational tools to address issues of data integration and data mining, in addition to traditional wet laboratory experimentation. She built a computational infrastructure to support NIAID, Malaria Host Pathogen Interaction Center, MAHPIC, and the DARPA HEMA project focused on host resilience to malaria. She is a joint PI of the NIAID Bioinformatics Resource Center, EuPathDB, a family of databases which integrate large omic and population datasets for eukaryotic pathogen research community. It currently contains data for over 300 organisms and has an average of 70,000 accessions per month. So that is a mouthful. But before I hand over the stage to Professor Kissinger, I would like to mention that Prof. Kissinger was part of our SAB, which is the Scientific Advisory Board for HA Bionet in Phase 1. And to be honest, we truly would not be where we are today if it was not for her contribution and immense support. So thank you so much, Professor Kissinger. Without further ado, please join me in welcoming Prof. Kissinger to give this talk. Thank you, Prof. Kissinger. Over to you.
Thank you. Okay, can you hear me okay?
We can hear you.
Great, thank you. I appreciate the opportunity to share a little bit of the research that I've been involved in for the last couple of years. I can get talking very quickly, so if I do, somebody please raise your hand or pop a note in there for me to slow down. Likewise, if technical difficulties kick in. So, as was mentioned in the introduction, I'd like to talk to you a little bit about some of the challenges and opportunities for the omics of infectious diseases, and this is from my perspective, but also I would like to then show you some of the tools that we've built to try and either take advantage of or to deal with some of the challenges that have been presented to us. So I'd like to begin by just sort of pointing out that when one works with infectious diseases, that pathogens are not model organisms, and they often present some pretty complex biological challenges. Often, even though we're trying to work with the genome or something omic related to an organism, we may not know its karyotype. It may not have a genetic system. If we're dealing with viruses, we maybe have quasi-species or bacterial panmixia, of the sharing of different genes in the population. You can have local biovars. Often pathogens have been known to hybridize. Some particular genera suffer from aneuploidy or polyploidy. They can have very complex life cycles involving numerous hosts. Very often you don't have a culture system where you don't have the ability to clone a pathogen so that you can get pure isolates that would allow you to develop cleaner sequences, to understand. They often have very large multi-gene families, which can make putting genomes back together again or understanding complex gene regulation of very closely related genes difficult. And they often evolve very rapidly and can have a complex population structure that's affected by their various hosts and geography. When we think about some of the opportunities, we're very fortunate to have tremendous advances in the next generation sequencing realm, so you can sort of sequence it all and then try and figure it out. We can get around some of the clonability issues or the culturability issues now with single molecule sequencing approaches. We can do flow cytometry to try and help sort various pathogens. We can actually, by sequencing it all, for intracellular pathogens, you can sequence the pathogen and the host cell at the same time and see what's going on and get that molecular snapshot. And then there are huge advances in transcriptomics, metabolomics, and additionally now understanding of the microbiome. And so all of these things sort of play into some of the complexities and also the opportunities that exist for working in the infectious disease realm. Now, one of the things that I like to think about when I think about bioinformatics and tools and trying to make all of this data visible is the fact that, you know, all of us probably have the experience of needing to book a flight or a trip or, you know, go to a meeting, wherever it happens to be, and you can log on to, you can pick your favorite sort of company, and I'm sure there are others in different countries around the world, where you are very quickly able to, with a pretty simple query, say, I need to fly from here to here on these days, right? And immediately you're presented with a number of options that might fit your criteria. And I just want to point out, these options come from competing airline, hotel, and car rental companies. And there are also competing sort of data mining platforms that let you look at this variety of research. And I think more importantly, I really am intrigued by the panels, the filters that you see over here on the left. So I'm going to amplify that on the next slide. All right. And you can see that you get everything from the site being able to try and provide you some advice, which it couldn't in this time, as to whether or not this will be the cheapest fare I'll see, to how many stops I want the airplane to take, when I want to take off and land, which airline I might want to fly on, how long I want my trip to be, how much money I am willing to pay, which company I'm going to be flying in, which airport I want my layovers. And there's an incredible number of options here that actually allow us to customize our search, right, on a variety of different data fields, ranging from the type of aircraft to the cities involved to the times involved, to allow you to find exactly what it is that you're looking for. And so I like to contrast this often then with the situation of what we would do in the biological disease world, all right, when we're looking at infectious diseases. Very often, researchers are looking for vaccine candidates or drug candidates or any number of interesting biological questions. And sort of the challenge is, why can we not approach looking at the data as easily as we were able to search airline flights, right? So to ask ourselves, if we're looking for a vaccine candidate, what might it look like, all right? And at this stage, we don't need computational tools. We need an understanding of the biology, right? So we might say, if it's a vaccine candidate, it will probably need to be a protein. It will probably need to be on the surface of the pathogen. So to get to the surface, you will need a signal peptide. All right. So necessary, but not sufficient to make it a vaccine candidate. So we're going to want a number of other filters, right? Might need a transmembrane domain or a GPI anchor to help keep it on the surface. We probably want evidence that the gene is actually expressed at the time that the pathogen is in the appropriate host that you're trying to immunize. And ideally, you'd also like protein expression to prove that not only is the gene expressed, but it's translated and the protein is present. You'd probably like to know even that the immune system sees that protein. And you can do that by looking at diversity in the target and seeing whether or not, analyzing the polymorphisms present, whether or not this seems to be under diversifying selection in an attempt to evade the immune system. You'd like to know that it's not present in the host. So to do that, we would have to check orthology relationships so that you can know whether or not, if you were to develop this as a target, you would have interference from the host, right? You'd like to know, ideally, that the host already sees this particular target. So if you were to look at sera, the malaria community has done this. They have protein microarrays, and then you can screen host sera against them to see which proteins are already recognized by the population. Again, all of these filters adding additional strength to the list of candidates that may be coming out that it might actually fit your bill. And so then the question becomes, how do we, as bioinformaticists, address what is a very, I would say, common set of biological questions that might like to be addressed. And so I think it's, I spend a lot of time thinking about why is there not the biological equivalent, of a travel search engine. And I've spent a lot of my career together with my collaborators and a very talented team of developers trying to think about that. And partly, the situation we're dealing with is much more complicated than airline travel, which despite the number of variables that we've talked about, companies, seats, cities, etc., it's still a defined population of characteristics. In the infectious disease realm, depending upon the disease, there are pathogens, whole pathogen research communities. There can be vector communities. There are the experimental and basic systems biology. There is host and host surveillance and epidemiology and any number of different sources and streams of information coming our way, all of which, in an ideal scenario, need to be integrated to help us try and address the challenges that we face to be able to try and ask those questions that I posed on our hypothetical example of looking for a vaccine candidate. So on this next slide, and I apologize, it's going to take a second for the pictures to come in. So some of the data types that we may have to deal with. And so you may have a genome sequence and its annotation. You may have blast similarities. You have protein expression profiles, comparative genomics, looking at the phylogenetic relationships, geographical distributions, cellular localizations, protein structures, protein expression, orthology analysis, RNA-seq, your Go functions, protein-protein interactions, chip-chip or chip-seq, pathways, host pathogen interactions, the immune system, some sort of mathematical modeling, perhaps on climate change or the distribution of hosts, for example, birds migrating and carrying various viruses around, like influenza. And so the slide is sort of meant to be a reminder of the complexities and the number of different data types that we're trying to deal with, that these are all data that we're trying to standardize, and the community is still working on those challenges, as well as utilize to be able to ask the kinds of questions that we would like to ask. And if you're like myself or most biological scientists, manual data processing is not very rewarding and it's a very painful process to go through in the attempt to try and find the answers that we're looking for. And so, and it's not been made easier by the advances in all the technology that allow us to sort of sequence it all, and that the amount of data that are available, and these are just sequence data and just in the NCBI data repository. And I'd just like to draw your attention to two features. So the blue line here is the number of GenBank sequences, and I can tell you that very beginning right here when BLAST was developed is when I started graduate school back in 1989. Right? And then the red line is the average number of users, and it's really quite amazing, right? That now the number of users of data has exceeded the amount of data, even though both are explosively growing, right? So there's a tremendous need to access the data, and there is a tremendous amount of data to be accessed, and this is just of one type, and this is just of nucleic acid sequence. So the situation is much more complicated when we think about trying to integrate all of the data. So you can ask the question, why is it so hard? I'm hoping that I've shown to you, for example, taking the airline or the travel industry, that it's not a lack of technology that can permit databases to talk to each other. The technology most certainly exists. But as I've stressed, the subject matter is different and much more complex. And I'd also argue, just sort of politically, the incentives are different. With travel, there's money to be made via the cooperation. And while with science, there are certainly advances to be made, corporations are global, but science funding, unfortunately, has borders. And often, in our particular culture, we're not rewarded or funded for playing together. And that creates barriers to being able to share data. And of course, as an academic, I fall into this category myself, making our own special terms and tools, coming up with our own special way to do it, is, again, sort of what we're rewarded for. And each time we like to think that ours is better than everyone else's. And so I think it's a combination of the subject matter and the environment in which we live, but I would also argue that it's possible to overcome these barriers. And so I'd kind of like to show you a few of the things that we've been doing as we think about these problems to try and make it easier to deal with data. And I really like to imagine a world in which data are easily accessible, in which the majority of them have been digitized, which means that you have the ability to access them somehow over the internet or computationally, in which we knew where and how to find the data. This is often a very, very hard challenge to overcome. Great if the databases can talk to each other, and in which we didn't need lots of file conversions. And so we have a world in which we have biologists with practical wet bench questions. I need all genes. I need to find all some proteins that have a particular property. Data scientists who are really good at managing and sorting data, and we need to have a meeting in the middle here. And so sort of with that in mind, I am going to move on to talk about, so if you were to think about the elements of an architecture that would kind of meet that, the approach we have taken is tools built by biologists for biologists, but we've expanded this to include clinicians, epidemiologists, and even to be able to support modelers and folks that we might not have traditionally considered falling into the consumers of data. And ideally, the tools would provide the ability for users to both mine and visualize their results, to implement their own sorts of search strategies, to save and share them, where you would have record pages where you have sort of encyclopedic compiled knowledge about a gene or a topic, and some kind of dynamic visualization that actually allows data exploration, right? So not just presented with a static image of here's what we know, but to let you actually say, well, what if, and play with parameters and change things. So I'd like to talk to you about a system of databases that I've been involved in for nearly 20 years. This is the Eukaryotic Pathogen family of databases. Currently, there are 327 organisms that are covered by these databases, and I've indicated in the numbers above the images of the different pathogens the number of genome sequences that are present for each of these. So we have 46 trypanosome sequences, only 1 trichomonid, 30 amoebas, 137 fungal sequences. Okay? And I will add, as I've put here in big letters down on the bottom, that we're also making resources now for host. We have human, macaque, and mouse, because again, disease is that interaction of the host and the pathogen together, and the changes in omic technologies are permitting us to study both now simultaneously, where you want to know, well, as the pathogen is doing this, what is the response of the host, simultaneously. So I want to take a minute to just sort of talk about the diversity of these organisms, because you may remember I began my talk by saying that pathogens are often quite different from model organisms. What you see here in the center around the circle is this is just a tree of the eukaryotic branches of the tree of life. And up here towards the top at about 1 o'clock, you can see the little girl jumping. So the epistoconta are the branch in which you will find humans, fish, and the fungi. And that's just this one small sort of purplish branch here. But as you walk around the tree, you'll see that with the exception of the plants, the majority of the diversity present in eukaryotes are actually protists. And the pathogens that we are making databases for are very, very different evolutionarily. They are not necessarily closely related to us or to each other, and they're as different from us as we are to trees. And this creates some very special challenges, and they each have very particular biologies for being able to take care of them. I've listed across the bottom the names of the different component resources that make up EuPathDB. So the first database that we made was PlasmoDB for Plasmodium parasites, and that's the agent that causes malaria. But we also have trypanosomes, Giardia, and host, and a database of orthologs. That's for determining when you have the same gene but present in a different species. For example, we talked about finding a vaccine candidate that might be present in the pathogen but not in the host. So what I would like to do then is just to talk about that for each of these pathogens, the data types are endless, and as the technology develops, new data come along. When we started, we were very happy to have a genome sequence, and if you were lucky, an annotation. But you can predict protein domains and relationships to others. We can now look at transcripts and their profiling in proteomics. You can have field and clinical isolates with their own associated metadata, for example, where they were collected geographically or what the patient might have been experiencing. We can look at any number of chromatin modifications, metabolomics, phenotypes. We can knock out genes. There's CRISPR. There's resequencing studies for population dynamics, et cetera, et cetera. And so any sort of data management system is going to need to be able to grow to face some of these challenges. So I'd like to just take a few slides and walk through sort of how we might, in the platform that we have developed, address a question sort of related to the vaccine candidate question. So here is the front page of PlasmoDB. I'm not going to spend a lot of time walking through it. And if you don't happen to work on malaria, I would just like to ask you to imagine your favorite pathogen or system that you're working on, and it might even be host. And I'm going to sort of just walk through here and do a search. So I might want to find all genes, which would be this first panel here in the darker purple. And I might want all genes that have a signal peptide. And I can say a signal peptide and in what organism. I said, well, I don't care. It can be in any Plasmodium species. And I can click on Get the Answer. And it will come back to me with a strategy panel up here at the top saying, well, I found 10,604 genes that have a signal peptide. That's obviously a very large number of genes that's too difficult to work with. And so much like we were searching for flights, we want to go through and apply some filters to try and narrow this down. And you can do this by adding a step. I will point out that those 10,604 results are displayed down towards the bottom of the page. And in the table in the middle, it's showing you how those 10,000 results are broken up by the different Plasmodium species that were present. If I add the step, a new window will open and it will say, what would you like to do? And I'm going to say I want to run a new search. I'd like to find genes. I would like to find genes that show evidence of expression. We'll say what kind of expression, and I can search for RNA-seq. It will show me the RNA-seq data sets that are available and have been entered into the system. I can pick one that says the blood stage. That means they would be present in the human host at that time. It will tell me, well, for that particular experiment, we have time points from 0 to 48 hours. And I can pick a particular stage. I happen to just say, well, at 8 hours that it's expressed, so early in the blood stage infection. And now, part of the power of our system is we get to say, how do you want to take the information that you'll get from this transcriptome analysis and combine it with your signal peptide search, your step one search for signal peptides. And I could say I'd like to intersect them. In other words, I'm interested in finding genes that have a signal peptide and are also expressed at 8 hours in the blood stage. And if I do that, then the strategy is modified, all right, to show me that there are only 165 genes that meet that criteria of both having a signal peptide and being expressed at the 8-hour stage in the blood cycle. And I can then add another step. And in this case, I might say that I want to find the orthologs of these genes in another species. Perhaps I'm working with Plasmodium vivax, where it's not culturable, so there's very little functional genomic data available for it. So I could transform that information, pick vivax, and say, now, well, of those 165 genes, it found 146 orthologs. Okay? And so in this way, we can continue to apply filters, or we can stop at a given stage and say we want to analyze the results we have. And I'll just point you over to the upper right-hand corner of the slide, where you'll see that you have the ability to save this strategy, to share this strategy, to delete it, any number of things that you can do with it. In this case, I'm going to go on and show you that we could share it with the URL. I could go down and I could add additional information by saying adding columns. Maybe I'd like to see the levels of expression, and I can add that column and I will see the graphs to be sure that I have retrieved what I think I have retrieved. I can also say I want to analyze these 146 genes, and I might go in and do a gene ontology enrichment or look at the metabolic pathways that they're involved in. And so if you pick the gene ontology enrichment, you can pick which of the gene ontologies you would like to look for. And then we will provide you with all of the statistics relative to a background test and multiple hypothesis testing for the particular genes that showed up in this particular study. So again, a way of helping the researcher to validate some of their findings. Okay, so in addition to analyzing the results, right, we can go to a particular page for any given particular gene, like the first one shown here on the list. And we could open up a page, and I'm not going to walk through all of these, but again, this is where I sort of mentioned that kind of encyclopedic content in which we could let you know everything we know about its gene, its annotation, link outs to other database, where it's found in the genome, associated literature, taxonomy, its orthology information, synteny, its position shared in the genome relative to others, transcriptomes, protein structure, etc., whatever data we have available to it, including immune reactivity. So then moving on, what I'd like to talk about and spend more time on today, because the things I've shown you today have been published previously, as the database has been around for quite a while. I just want to tell you about the ways in which we're growing with these additional data sources as they become available. So we've really begun to say that, you know, once upon a time there was only one genome sequence and users would come and they would look for their gene. Now we have thousands of genome sequences for some pathogens, and what's really important is the data about those sequences. Where were they collected? What is the nature of, you know, were they more pathogenic or less pathogenic? Are they resistant, right? So the metadata, the data about the sample is very, very important. And so we have expanded extensively to include additional metadata and to allow researchers to find samples and experiments based on mining that metadata, which has obviously involved a lot of ontology development. We're created workspaces where users can bring their own data that are not present in the database, analyze them through Galaxy workflow pipelines. I'll show you an example of that in a minute, and be able to see the results in the context of our database without sharing it with anyone else or us. You can analyze a list of genes. Perhaps you've done an experiment and you found a number of genes that fit some criteria and you want to know whether or not they're enriched for some particular process or pathway, as I just showed you a minute ago. We're providing curation now for kinetoplastids and fungal, I apologize, that is misspelled, genomes. And we're having to deal with new data types like systems biology data that are evolving from systems biology projects like the Malaria Host-Pathogen Interaction Center. Okay, so this is an example of, and I apologize that it's busy, but I understand the slides are being made available. We've created a Galaxy cloud environment. This Galaxy environment can be linked to from any of the databases. The example here is just from the fungi. But what we have done in this Galaxy environment is to preload all of our reference genomes and provide, and so we've shared with the users, a number of workflows that allow you to either map RNA-seq data to the reference genomes or to map that data and then look for differences in expression if you have a time series or different experimental conditions. We also allow users to map reads and call sequence variants, and we can handle single or paired end data from a number of platforms. Users can go in. This is free. You can upload your data. We don't see it. It goes straight to the Galaxy environment. This is just an example of some of the workflows. But then what we've done, and these are all found under that Analyze My Experiment tab outlined in sort of the gold color up on the top header, is that once the results are done, not only could you download results to your own computer and continue on with your studies, but we also allow you to bring back data tracks, as shown here across the bottom, where you're looking at some mapped reads. This is a bigwig file shown back to the genome, where you can bring them back into the database and display them in the context. You can load the track and see it in the context of the annotation and or any other data tracks that are publicly available in the database to analyze your sequences. So this has been a very useful feature. We have 810 registered users of this service, and last month they ran over 7,000 jobs. So it's very heavily used. We've also, this is through efforts that are not funded through the NIAID contract, but an independent effort set up, built a microbiome database that can allow you to go in and mine the metadata about microbiome experiments. So now you're not just interested in looking at taxon abundance within samples, but you're interested in comparing samples that may share similar taxon abundances or come from similar environments. So for example, you might want to know, if we look at this last question, what's the impact of delivery mode, vaginal or cesarean, and the mode of diet, formula versus breastfed, on the gut microbiome? And this has the ability to cut across different data sets to be able to answer that question because they've all been processed simultaneously. You will have all of the normal types of parameters that one would like to look at for microbiome studies. And in particular, if I look at that results of baby's delivery mode, you'll see that there are graphs here where you can have cesarean is in the orange, vaginal is in the green. You're looking at distributions of different bacteria. But what's so nice is that you can go up and you can click, and instead of just delivery mode, you could just, with the click of a mouse, click what they were fed by, and you would have new plots, and then there are tools that you can play with and allow you to dynamically interact with the data. So you can see, you can play the same buttons up here. You can analyze results, and you can go in and work on them. Here's the selection of picking child's diet, and just, you know, with the click of a mouse, you can then see breastfed, exclusively breastfed, or milk formula. Okay. The next tool that we've developed, and again, these are all using the same back-end hardware, is developed for clinical epidemiologists. This is an entirely new audience of users that will require a different interface to be able to look at their data. But instead of now asking for all genes that have particular properties, we're now interested in all participants of a study that might have had particular clinical outcomes or faced particular circumstances. And this is a project that's funded by the Bill and Melinda Gates Foundation. And so I would just like to show you some of the features that we've developed. Now, as I mentioned earlier, we've had to do a lot of ontology development because in order to compare subjects within studies, we need very controlled vocabularies, just like we do when we talk about a gene or a transcript or a protein. And so we spend a lot of effort collecting information and developing standards for the types of information needed to be collected when you're dealing with human pathogen or vector genomic sequences and data. For example, you can have a participant, right? That participants live in dwellings, and dwellings have geographic locations, but that patient also can have a visit outlined in red up at the top, and that visit happened in some location. That visit might be part of a study. That study might be one of the International Centers of Excellence for Malaria, or might be an H3 Africa study, right? Dwellings might have mosquitoes, right? And mosquitoes can be related to entomological measurements. And so we've really gone after trying to capture some of the complexity of these studies. And so the data that I'm going to show you today, I have permission to show you. I have obscured any personal details because some data, of course, cannot be shared. The Bill and Melinda Gates Foundation contacted us about trying to make resources because they've funded a number of studies, and the GEMS 1 and 1A studies and the MAL-ED studies were specifically looking at causes of diarrhea in the early years of life and the particular consequences or associations with particular either disease outcomes or stunting in growth. And so the MAL-ED study is much longer than the GEMS study, but the papers are shown here if you'd like to follow those up. And so the MAL-ED studies are not yet public in the databases. The GEMS studies are about to go public with some data release caveats that I'll show you in a second. All right, so first thing I'll show you is this is the front page of ClinEpiDB. You'll notice it looks very different than the front page of the PlasmoDB that I showed you. We very much try to tailor things to audiences. We're talking to epidemiologists and clinicians. The main components of this system are tiered data access. In other words, some data, as we know, are not sensitive and can be accessed without personally identifying anything. We have no relationship or knowledge or way to get back to any individual study subjects. But some information, even if not identifying, might be considered sensitive or subject to particular regulations in particular studies or governments. And so there is a tiered data access system for individualized data, but not for the summary data. So most of the data I'll be showing you are summary. And like our mining or filtering for genes in our gene system, there is a search wizard that helps you find participants from these studies, and a strategy system along with results, the ability to explore and interact with your data, and download it if it is available for that study. So just walk you through it. Here we can explore it with the wizard. If you can see, we can walk across the top here. You can look at geographic regions, personal characteristics, some observations, case control studies, and some result options. You can see we started out with 22,567 participants in this particular study. If you look down below, what we've done that's a little different than in our molecular mining systems is that we're immediately showing you what countries the participants are from and how many records are there. So if we were to go in and ask for participants from a country that are not represented, of course our results immediately would be 0, and we wouldn't ask for that. Here we're going to filter down. The data are being shown to you, and you can select on them. So if we were to take these participants and then only look at those who are male by location, you can see that the full length of the bar and the distribution all the way out to the end of the gray is showing you how many there are. So for example, in the first line, in Nepal, there were 130 households that have male by location out of 240 total. So now the pink is showing you the households that have been selected relative to the total that existed. And so this is a very nice way to filter. And then you'll see over here on the left, we can filter by any number of characteristics, from how many rooms there were in the house to the flooring material, the elevation, the latitude, the longitude, etc. But again, not close enough to identify any particular household or individual in this case. And so we can go through and we can explore data also by histogram. So this is looking at male by their weight for age. And again, you can see the full distribution of the whole data set in gray, but because we have selected only males, you can now see the distribution of this particular characteristic in pink. And the nice thing is that we have the ability to use our mouse to select these things very quickly. So here's an example of maybe we want to find all children that enrolled in their second year that are in their second year of life in the study. And so we can pull up the histogram for personal characteristics that's showing the age, right? And then we can just drag your mouse over this part here from 12 to 24 months, and it will immediately grab that subsection of participants, which reduces it down to 7,587 participants from the total that are available. So a more interactive way of doing your, performing your data mining. And then if you needed to see details for an individual, again, we put a lot of effort into thinking about how to visualize a lot of information very quickly. And this really came from a lot of feedback from the community that we were serving. And so here is a generic record, all right, for an individual, in which we can see over the entire course study, which here is at 700 and some days, okay, where we can see plots of their weight, their height, or their weight for age scores, as well as indications shown in the navy blue. You can see the rectangular boxes below, indicating when they had particular episodes of diarrhea. You know, and this latter box there lasted for 15 days. So you can see there were a very large number of diarrheal episodes in this infant's life. But the amount of information that you can get at a glance is really important. And similarly for malaria studies that we are been working with for the isomers, and these data are public and available in the database. This is some results that were returned by a strategy search, where you can look at individuals and you can see the red dots. And so these bars on the right-hand side of the slide are showing you episodes of malaria that individuals had, and this is a 1, 2, 3, 4, over a 6-year period. I mean, it's really amazing. And to be able to visualize and so quickly see patterns. So, for example, that we can see in the latter 2 years, there is much less red in the results returned from this particular search. Okay. As I mentioned, we've put in the search strategy system for being able to mine participants as well. So in this case, we can do a search for, in this case, all children, right, that had a very good height. I believe this is the height for weight measurement, that for many times, followed by having at least 5 of those measurements that was now less than a -2, and that was after being positive for having a microbe and having an episode of diarrhea. Right? So you're looking for children who were doing very well, but then following episodes of diarrhea in which there was an infectious agent involved, then we're measuring more poorly for this particular measure. And so you can go through and you can do each of those tests where they had to be, microbe positive, they had to have diarrhea, they had to have been seen, etc. And we end up with 26 participants that fit this criteria. And then just as we could have lists of genes, you can have lists of participants, and you can look across their measurements. And again, we can see this dip here in the measurement. We can have results tables, again, where you can look at many, many individuals and you can add columns and learn additional information about them. For example, you might want to then add, well, what was the circumference of their head, or what was the mother's age, or the mother's body mass index, or has there been a previous infant death in the family, etc. And you can add this information just with a click on the fly. So again, trying to make the results exploration much more dynamic and informative. All right, so I wanted to start to wrap this up, and I kind of want to bring together some of the different things that we've talked about. And the focus of this talk was thinking about the complexities of infectious diseases in the omics era. And I think, as we all know, because you're involved in the H3 Africa projects, the development of disease is a very complex phenomenon. And it can involve, this is an example for malaria, but it can be any particular type of infectious disease where you are going to have pathogen factors that are going to contribute. You're going to have host factors that are going to contribute, and then there's going to be geographic and social economic factors that are going to contribute. And all of these different streams of information coming in will lead to a particular clinical outcome. And so, as we think about this being data managers and data miners and sort of things that we do, the challenges are immense. Discrete data, a particular data type, a height, a measurement, a temperature, or a gene expression value, they're relatively easy to do. Even, we have a very large nucleic acid database. They're nucleic acids. You can put them all together. And so we have databases that are really good at capturing individual particular types of data. What is really hard to capture is an experiment, all right, where you have to bring all of these different data types together, right? And depositing an experiment is really different than depositing a data type. You need the big picture. You need to know why the experiment was done. What were you trying to measure? There could be all kinds of information about patients or animals or sources, all kinds of kits and machines and protocols, various types of results, some of which don't have a database to be deposited in. There were all kinds of statistical procedures applied to normalize or deal with noise in the data that you need to know how they were done. And while we have databases for these discrete data types, we don't have databases that capture this kind of information, like time series and drugging schedules, diets, animal caging or instrumentation, all of this clinical data that we want to be able to correlate, to be able to bring together in sort of one pie. I think of these as slices of the pie, to have a more holistic view of everything that's going on as we try to develop the tools that are going to help researchers in our fights against these diseases. And so we put a lot of thought into trying to develop some tools that will help facilitate this. One of them is our host-DB for looking at the host-pathogen interaction. There is not an equivalent host database for human that I have shown you out there, and so we've created one for human, mouse, and macaca mulata. Some additional hosts are needed, but the only data that you're going to find here are ones where there's a host pathogen data set. So for example, there might be an analysis of a human proteome infection with 4 different strains of Toxoplasma gondii, or looking at the Giardia secretome when they're in human intestinal epithelial cells. All right, so again, we're just trying to break down some of the barriers that make it hard to study disease in a number of different contexts. And finally, I will tell you the things that we're sort of thinking about is ways in which we can bring it all together. Because you need epidemiological and clinical studies, you need molecular studies of both the host and the pathogen, and we know the microbiome influences both the host and the pathogen, of trying to bring in all these different sort of data types to be able to study the host-pathogen interaction. And so we're very, very hard at work thinking about ways to enable cross-informativeness across these studies. So for example, in the International Centers for Excellence in Malaria Studies, their data has been deposited in epidemiology, but they also have collected both mosquitoes and parasites, and we have molecular information about those agents from these studies that could be located in additional databases like EuPathDB. And so how is it that we can link the information from these different aspects of a study together? So we spend a lot of time thinking about that. And so I just kind of want to finish up by showing another slide that David Roos generated that I think is really important, and that is, we have, it's shown here by these different covers, titles, we have genomes for the hosts and the pathogens very often. We have a number of tools, and we really have a tremendous need to develop drug targets or vaccine targets, and we all really need to find a way to work through this data and come up with the tools to come up with the sorts of breakthroughs that are needed to help us deal with them. So I'd like to finish my talk by thanking the team that is involved in making this possible. It's a very large team. There are nearly 40 of us now, and I really also have to thank the the community of data providers, and the study participants and the feedback from researchers that have helped us to make the tools to where they are right now, and to my co-PIs in these projects, David Roos at the University of Pennsylvania and Christiana Hertz-Fowler at Liverpool, and to our funders. I will stop there and take questions. Thank you.
Thank you so much, Professor Kissinger. We had a bit of some technical glitches in the beginning, and I'm glad that it all worked out. So thank you so much for your time and such an amazing and interesting talk. As I said before, we are really grateful to Professor Kissinger because she was actually part of SAB in phase 1, which is the Scientific Advisory Board. But for now, we're going to focus on this talk. So if you have any questions, please raise your hand. What you can do is go on top, on the top bars, on the top tab, there's something that's raising its hand. So please, there's like an icon of a human being raising its hand. So please press it, and then I'll be able to give you rights to ask a question, and Prof. Kissinger will answer your question. So are there any questions? Victor?
Yes, sorry, it just took me a second to unmute my microphone. Jessica, this was a really nice talk, really appreciated it. And you presented a whole bunch of really nice data exploration environments, which obviously should help people get new insights into the data. And my question is, do you have any specific example where having these tools, these exploration environments in hand, has led to insights into the data that would not have been possible otherwise? So do you have practical examples where researchers looked at the data using the tools that you developed and came up with new insights or new testable hypotheses?
Most of them are offhand or seeing snapshots of our pictures in various presentations and talks as you go to different meetings. I do very much remember in some of the earlier days, just the ability to have the genome sequence available and mine it. I heard a number of anecdotal stories from study sections where people had sent in an update saying, oh, forget AIM1, we were able to find the gene, by doing data mining. That's already done now, So what I think it's doing is facilitating the speed at which people are able to find things. ClinEpiDB just went public about 2 months ago, and so I don't have stories yet where people have actually been able to take that data, or we haven't yet enabled the platforms that allow people to connect ClinEpi data with EuPath data to facilitate this kind of next generation of breakthroughs that we're hoping can happen. So I don't have studies from that. It's a very good question. There are numerous times you're sitting in the audience at a presentation, and they were like, "Well, we were curious about this, so we went to the database and we found this, and then it led to a whole story that people are telling." So it definitely is facilitating research. We should really keep that top 10 list of major discoveries. If we know about them all, because we don't. Databases are often taken for granted, as you may know.
Oh yes.
But this is it's a very good question. I do think what I can tell you is, you have, these are funded with taxpayer money, right? And so if you're just making tools, you don't want to make them if they're not being utilized. One slide that I took out was our usage statistics. So if you do a Google Scholar citations, because people don't always cite the paper, but they will mention that they went to PlasmoDB or TriTripDB to do a search. We have over 16,000 Google Scholar citations, and when we look at, we never watch exactly what people are doing in the database because that's unethical, but the browsers, the servers always track IP addresses. We can say that the same people come usually like 3 times a month and stay for more than 30 minutes. And that's how we like to count users, as opposed to robots. And so we're accessed by like 70,000 users. This is after we remove robots and stuff per month, but with the average user coming at least 3 times and staying for 30 minutes. Which would indicate that there is something of value here, or they wouldn't come back, if that helps answer your question.
Yeah, no, that does answer my question. Thanks very much.
Thank you so much, Victor, for that question. Is there any other question? Again, please raise your hand, then I'll give you the right to speak. So go ahead. If you have a question, please press the icon on top. There's a human-looking icon raising their hand, so press that, and then I'll be able to see if you have a question. Okay, I'm gonna give it a few more, I think 1 or 2 minutes, and if there are no questions, I'm sure that all of you got the ad, the MailChimp ad that we sent out, which has Prof. Kissinger's email. So I'm sure if you want to engage further on this topic, Prof. Kissinger wouldn't mind to engage with you. So if you don't have any questions now, I will give it a minute or 2 now to see if any questions come up. But if not, I'll encourage you to engage with her via email or other platforms.
Well, I would just like to thank everyone for their attention in this seminar. I think it's very hard to attend a seminar remotely, but I'm also very, very thankful that we have this technology and that the H3 Africa platform makes this a available as a way of sharing information with each other. It's been a pleasure to present to you.
Thank you so much. Like I've said this before, but I'm going to say it again because I'm really, really grateful for your time and such an interesting talk. It was, luckily I recorded it, so it's going to be available on YouTube for others who were not able to attend, but also for us who want to go back to this talk to obviously hear things that we might have missed. So we really appreciate your time and your effort. Thank you so much, Prof. Kissinger.
Thank you.
Sure. I think on that note, I'm going to close the room. For those of you that are attending, once I close the room, you're going to get a link which has a survey for you to answer questions on our webinar series and stuff. So just look out for that. And Prof. Kissinger, thank you so much and enjoy your day further.
Thank you. Bye-bye.
Okay. Bye, everyone.